Okay, so hello everybody. My name is Sabina Vitola, and this summer I had the opportunity of working with the LOCERT lab in the biophysics department of here at UMD. My mentor was Nick Minona, and my project was in simulating spontaneous calcium oscillations in astrocytes. Okay, never mind. How do we switch this on? Okay, we did it. Okay, so after all this physics, you probably didn't know that you were going to get a bio lecture. So let me give you some background. First of all, what are astrocytes? They're actually the neighbors of neurons. They're a type of cell called a glial cell, and it is found in the central nervous system, so the brain and the spinal cord. They're active components in the brain, so they communicate kind of akin to how neurons do in networks, but their communication is much slower, and it's through calcium waves, so it's chemical communication. I have a diagram of the astrocyte. It's the blue one. Actually, there you go. That's the astrocyte. And on top of it, we have two neurons, and they are communicating. So they have that synapse in the middle. And the arrow is showing you probably the most important role of astrocytes, which is they are supporting the propagation of information through that synapse. And this is called the tripartite synapse. Okay, so since we have this communication, we have a network. So essentially, calcium waves can propagate intracellularly, which is in one cell body, or intercellularly between two different cell bodies or several cell bodies. And they're triggered by various mechanisms. So we have various outputs and inputs coexisting. The intensity of calcium events can vary, and they're slower dynamics than neurons. That's, again, something that's very important. So right here to your right, we have a video of a stellate, which is a type of astrocyte network, and it is firing. So this is what I call when I say calcium events. This image was, or this video, this trace was taken by Nick Minona, my mentor. If you think it's weird that his picture is there, I agree, but he told me to put it there, so there you go. And we can actually see what I'm talking about here. So we have these calcium spikings that are targeting one cell specifically, but you can see that it's propagating throughout the whole network. So these will be calcium events, and that's what I'll be talking about for the rest of the presentation. This video is actually 10 minutes, but it is a 10-second simulation. So that's why it's much quicker. It's flashing really quickly. But I just want to highlight the time scale to tell you that it's much slower than neurons. Okay, so this is biophysics after all. So how do we go from all of that bio to physics? Essentially, what I did this summer, or my project, was looking at computational and mathematical models of calcium events, or these calcium waves. So there's different ways of doing these models or looking at these models, and I'll be only talking about the intracellular. So that, once again, is within one cell body or one astrocyte. Calcium waves, we will be focusing on 3 different variables. We have the calcium concentration in the cytosol, which is in the cell body, and that's denoted by X. We have the calcium concentration in the ER, or the endoplasmic reticulum, which is an organelle in astrocytes that stores calcium, and that's denoted by Y. And then we have IP3 concentration, which is IP3 is a messenger molecule, once again in astrocytes, and that's denoted by Z. That IP3, I will show you in the diagram later, but that IP3 actually triggers the release of calcium, and it causes these waves. But once again, I will talk about that a little bit later. These cells are regenerative, which means that one cell can trigger other cells within the cell body, and they have the potential to propagate themselves the network. And they can be triggered either through neuro and neurotransmitters, so other cells signaling to the astrocyte, or they can be spontaneous. So that basically what it means is that there's mechanisms within one cell that cause these waves to start, and that's the one that I will be talking about. So specifically, the model that I will be referring to is the spontaneous calcium events in astrocyte model, and it's based on a paper, 2008 paper, and it had these 3 coupled differential equations. So the first one is the X system. So once again, that was the cytosol, the calcium concentration in the cytosol. So we have that calcium going in. The terms in red are the ones that are leaving the cell. So calcium leaving, and then the green is calcium entering the cytosol. Then after the calcium is inside of the cell body, it flows into the endoplasmic reticulum. Once again, we have green going in, red going out. And then our third system is the IP3. And so once that calcium, the other thing that calcium does when it's in the cytosol is that it stimulates the production of IP3. And IP3, once again, is that messenger molecule that binds to receptors in the endoplasmic reticulum and causes calcium to get pumped back out into the cell. So that is a positive feedback loop. That's what that's called. And this is what causes these regenerative calcium events or calcium spikings that I showed you earlier. So basically what this model does, it helps us with the understanding of pathways that are used by calcium to enter and exit the endoplasmic reticulum. And this can help us understand how that dynamic behavior of calcium affects astrocytes. And essentially what is happening, I just want to highlight exactly what the model does. The changes in the concentration of the cytosol are very specific or very, they can be triggered by really small mechanisms. And so that's what causes those events. Okay, so for my summer, my job essentially was to use those couple differential equations to create a graphical user interface. So I used MATLAB App Designer to create COSTIS right here. COSTIS stands for Calcium Oscillation Stimulation Tool for Astrocytes. So let me show you a video of what it does while I talk about it. Essentially, I use these systems or the variables that I talked about in the differential equations, and you can manipulate them to see how those parameter changes change the chemical behavior of the astrocytes in the three different systems. So that's calcium in the cytosol at the top. We have calcium in the endoplasmic reticulum, and we have IP3. As you can see, as I'm changing one parameter, because I can't show you all of them, I don't have time for that. But the parameter is the rate of calcium flowing from the cytosol to the ER. We can see how that behavior goes from oscillatory to non-oscillatory. And that's great to see, because if we kill COSTIS can come back to life, that digital twin can come back to life. But if we killed an astrocyte, that would happen. So that is the value of this project, essentially. Some applications of COSTIS is we want to use some function fitting techniques to actually take those experimental calcium trains, this is the one that I showed you earlier, and match it to the model in COSTIS. And so how do we do this? Essentially, the first thing we would have to do, or I would have to do, is constrain parameter ranges based on existing experimental evidence. So we want to make sure that it is congruent with the biology. And then after this, we would find, hopefully, parameters of the model in COSTIS to uncover probable biophysical mechanisms to explain the differences in the experimental traces. So we're reverse engineering what's happening with the cell and hopefully find some explanations of what is occurring with those calcium waves. So the value of my research was modeling, my modeling and analysis tool can help us unveil key insights about astrocytes behavior and deepen our understanding of the astrocyte neuronal network. This project, or COSTIS, will allow us to use experimental data showing altered astrocytes, and this can be due to developmental stage or even injury, to create a modified model that will provide us with potential explanations for these behaviors on a cellular level. So essentially, all that to say, models or projects like these can help put us a step forward in understanding cognition itself, even if it's a small step forward. Special thanks to Vitola and my mentor Nick and Dan Serrano.